Hello, everyone. Welcome to today's webcast, Automated Analysis of Purity and Packaging of Gene Therapy Vectors and VLPs using Transmission Electron Microscopy. I'm Rita Peters, Editorial Director of BioPharm International and the moderator for today's event. We're pleased to bring you this web seminar presented by BioPharm International and sponsored by Vironova. Vironova is a Swedish biotechnology company providing comprehensive hardware, software, and services for the analysis of nanoparticles. They specialize in electron microscopy with a 10-year track record of services to the pharmaceutical industry. To find out more, you can visit their website at www.vironova.com. I have a few important announcements before we begin. This webcast is designed to be interactive, and I encourage you to ask questions during the event. You can submit your questions by typing them in the Q&A box, which can be found on the right-hand side of your screen. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. Please note the resources provided by our sponsor that are located in the green resource widget in the dock at the bottom of your screen. And should you have any technical problems viewing or hearing the presentation, you can click on the question mark help widget in the dock at the bottom of your presentation window. Now I'd like to introduce today's speakers. Josefina Nilsson is the head of Electron Microscopy Services at Vironova CRO. She has a doctorate in virology from the Karolinska Institute. Since 2007, she's been head of the company's specialized electron microscopy services for biopharmaceuticals, shaping the company's services and solutions related to viral particle characterization. Vironova also offers innovative hardware and software solutions for advanced electron microscopy image analysis. Martin Ryner, Manager of Strategic Development at Vironova AB, is one of the pioneers behind the company's software inventions for analyzing and quantifying viral structures in electron microscopy imagery. At Vironova, Martin has managed system development and techniques for data mining, machine learning, and viral expression measurements. Before joining the company, he developed e-commerce models for the retail industry and control systems for industry automation. He has a solid foundation in applied mathematics from the Royal Institute of Technology in Stockholm, Sweden. So thank you both for being here today. Josefina, I'm going to turn the presentation over to you.
Thank you very much, Rita, for the introduction. So today, Martin and I will talk about automated analysis of purity and packaging of gene therapy vectors and VLPs using transmission electron microscopy. However, this approach can of course also be used for other nanoparticles. First, let me give you a short introduction to Vironova. Vironova is a Swedish company. We are located in Stockholm. We do offer unique services for nanoparticle characterization and viral clearance testing, together with hardware and software for advanced electron microscopy analysis. To be able to do this, we have, and I would like to emphasize that we need to have experts in interdisciplinary fields, like electron microscopy, image analysis, programming, mathematics, and structural biology. So by combining the expertise with our in-house developed software, VAS, we offer analysis of nanoparticles as a service. However, after many discussions with our partners and clients, we understood that they would also benefit from being able to do some of these analyses in-house. Therefore, we, together with the Long Instruments, have developed a tabletop transmission electron microscope, which we call the MiniTEM, which you can place in the ordinary lab facility. So why use electron microscopy at all? That I would say is because particle characteristics like the size, shape, purity, and the overall sample morphology, and I will come back to that, of your product can often be directly correlated to efficacy and safety. Electron microscopy is a great technique to really get to know your product, to understand your design space. Like what happens if I change my formulation or purification process in this or that way. So for all of you that are working with, for instance, viruses and proteins, you know that the process to get your product is quite complex. And there are a lot of parameters and steps to keep track on, like aggregation due to unsuitable up or downstream conditions, the trade-off during purification to get as pure product as possible without losing too much material. And of course, to make sure that your particles keep the right morphology and are intact over time during storage, for instance. And I would like now to come back to the overall sample morphology, as I think it's a very important parameter. For years now, we have been working together with clients within the vaccine and gene therapy field. And independently what kind of particles they are working with, it always comes down to find a way to characterize the entire sample, a way to understand the overall sample morphology. What kind of structures can be found in the sample, and what is the relative level of each structure? The tricky part here is to find a robust method to classify the structures that you find in your sample in a way so that you can compare the results from one batch to another. And when you can correlate a specific structure or even a specific structural footprint, as there is often more than one structure within the sample, to the wanted biological effect, then you have created a very powerful analytical tool. To make it a little bit clearer, I would like to show you an example of what we did together with the client just recently. The sample analyzed in this study contained of a couple of different structures, which we could classify by analyzing the size and the shape. The most frequent structure was quite small and almost spherical. This was also the particles responsible for the wanted biological effect. There were also some larger structures with different shapes, but these seemed not to affect the efficacy nor the safety, so these structures were acceptable as long as their concentration was not too high. Then there were also some elongated structures with similar size as the small spherical particles. And these flattened particles seemed to decrease the wanted biological effect, meaning that this was a structure that the client really wanted to monitor. So here we were able to classify the particles and set acceptance criteria for the two most important structures, giving the product a structural footprint of an accepted as well as a non-accepted product. So there are a lot of parameters that can influence the quality of the product and which can be monitored using electron microscopy and image analysis. Therefore, I would like to give the word to you, Martin, to tell us more about the MiniTEM system and how it can be helpful with all the automation that has been built into the system.
Thank you, Josefina and Rita, for this introduction to the setting in which we operate. It's great that so many are attending this webinar, and we're really excited to talk about how we're taking on these analytical challenges. Now, in order to gain the insights we have been discussing so far, we decided to build an automated transmission electron microscopy screening and analytical system so that we can investigate samples with as a stable and robust result as possible. First, some basics. The MiniTEM is a highly automated transmission electron microscope that provides a spatial resolution of about 1 nanometer. This is sufficient for most applications that we have been talked about so far. It has all the tools available for easy exploration of samples, as well as unsupervised screening and sample analysis. Because of the automation, the instrument can be operated on a generalist level, which makes it accessible for more personnel. The instrument can be placed in standard laboratory settings with no extra requirements of mechanical or electromagnetical interferences, which is usually the case when acquiring a full-sized electron microscope. Installation and commissioning takes a few days. I'm now going to guide you through the automated procedures in which the system works in order to collect and provide meaningful and accurate data of a sample. And we're going to discuss how this data and output can be representative for the sample and ultimately the product being analyzed. On the slide, you see a representation of a standard 3.1 millimeter grid, which is used to place the sample upon and subsequently mounted into the instrument. To investigate sampling variability and preparation variability, it's a good idea to investigate different areas on the prepared sample grid. 5 or 9 areas are usually enough to investigate variability or trends throughout the grid. You can tell the software to visit pre-configured waypoints within a certain pattern, or you can manually, based on a low magnification assessment, tell the software which waypoints to investigate. The system then screens these areas automatically using the inbuilt operational automations. Each waypoint will be visited and each area will be imaged in a structured way. This would take a few hours if done manually, which for an experienced expert would not be an attractive task. The system moves from area to area and collects images at each area based on the user configuration. Each image is processed and particulate matter such as the primer analyte, but also undefined structures of interest are registered and accounted for, so that we can perform automated classification, giving a condensed metrics of presence and relative concentrations and distributions of, for example, size or integrity of the particles. Since the preparation procedure of samples can affect the particle integrity, both stain selection and stain quality of grid areas are critical factors for accurate virus particle integrity measurements in transmission electron microscopy. Here we have 3 images acquired with MiniTEM show representative grid areas with different stain quality of adeno-associated virus AAV samples. On the left, we see a grid area with poor stain quality where the stain has been dried out. A dried area is whiter compared with the other areas, and the particles lack the typical stain halo that is observable during optimal staining conditions. When we scan for viral particle integrity status in these areas, we see a clear correlation between stain quality and particle integrity, which is an unwanted dependency. However, over a certain threshold of stain quality, the results stabilize and a comparable measurement between samples is obtainable. As a comparison in time consumption of performing this analysis, we have sample preparation taking about 15 minutes. Microscope usage takes in the MiniTEM about an hour, which is automated, leaving the analyst to perform other tasks compared with a manual operation of a few hours to collect enough images. The online image analysis takes about an hour per sample in the MiniTEM, providing about 60,000 registered particles. This is not at all a task that is feasible to perform manually, but extrapolated data shows that this would mean about 2 weeks of manual labor. Sometimes you are working with structurally quite complex samples, like for instance the influenza vaccine. Here, you often have more structures which needs to be classified and correlated to their biological effect. In these samples, you often find spherical particles, elongated particles, broken particles, and it's often that the more elongated particles are broken in the ends. And we have background debris. You can make the list as long as you want or as you need. And sometimes you need to go deeper into the structure to get a good classification, looking into the fine structural details. Here to the left, you have a nice looking influenza sample with nicely decorated particles where you can clearly see the spikes on the surface. However, to the right, the sample has passed through one additional purification step, which seems to have shaved off most of the surface proteins, wherefore most of the particles looks naked, and you find the spikes as free entities in the background. So while the product to the left has good biological activity, after one more purification step, it has lost most of it. So for influenza and lentiviruses, it would be a good idea to monitor the presence of surface proteins. For adenovirus, you might need to analyze the fiber in the five-fold axis. And rotavirus, you might want to make sure that you still have all three protein capsids. To deal with these localized features practically in the MiniTEM, we would start with detecting all particulate matter in the images down to a specific size threshold. In this image to the right, you see most particles of interest detected with the blue line surrounding them, but which are of different kind and of different biologic relevance. Also, we see a detailed debris in the background, but we will disregard from this right now. When we pinpoint localizations of details or contexts surrounding each particle which represent a specific and biologically relevant structure, these feature proposals can then undergo a robustness and generality assessment before being deployed. That is to check that the details are specific for a certain particle type and that we have separability of these unique features above the noise level. In the image on the slide, we have red particles that have the same structural features, the green that varies a bit from the reds, and there is a recurrent structure marked as blue surrounding the VLPs, which contains localizations of protein spikes. When this analysis is done, we can produce high-performance fast and fixed features which generalize these findings. For instance, we pinpointed that the surrounding band around the particles, and in these regions we can find the protein structures that separates what in these images are believed to be exosomes from influenza VLPs. With the different classifications in the middle image, green for VLPs, red for the exosomes, and yellow for something in between. Condensing this data, we can then end up with relative particle concentration estimations in the sample. Usually, a surface area-based weight metric is more appropriate than a particle count method because of the more usual surface-to-surface biological interaction. So, in conclusion, it's a good idea to track and have a clear view of the morphological footprint of your product, as it can be both reveal deviations hard to detect or to pinpoint by other analytical tools, and as it will also provide quite unique design space parameters. So keep track of the level of intact particles as its identification of the efficacy of the product. Keep track of the level of broken particles as it is an indication of the stability of the product. Keep track of the level of particle debris or host cell debris as they have the tendency to be nucleation sites for aggregation and unwanted immune responses. And keep track of the level of aggregates as it will induce unwanted immunogenicity. So back to you, Josefina.
Thank you, Martin. I will now focusing on how to analyze the adeno-associated virus particle, as we have seen a growing interest in using AAV particles for gene therapy. So let's go back once more to the overall sample morphology. Regarding the AAV product, we know that it is the nice, spherical, intact AAV particle that gives the wanted biological effect. In the image to the left, you can see these nice-looking AAV particles. The background is rather smooth, meaning that the product is quite clean. Not much debris can be seen. We can, however, see one unwanted structure, which is the proteasome, seen as a donut or barrel-shaped structure depending on the orientation of the proteasome on the grid. In the middle, you have an AAV sample with much higher background, meaning higher concentration of debris. You can find the proteasomes also here, as well as larger aggregates around 500 nanometer. And on the right-hand side, you have a really nice-looking AAV sample. Not much background, but we can here see structural conformations of what we call the duplets and the triplets. You can, of course, argue that these structures might be due to the sample preparation using negative staining technique, but we could also see them using cryo-electron microscopy and can therefore expect these structures to be present also in solution. Analyzing an AAV sample using negative stain, we can clearly distinguish between the intact form and the internally stained non-intact form, as well as measuring the ratio of doublets and triplets in the sample. However, as Martin stated before, you need to make sure that you are analyzing areas of the grid where the particles have been nicely embedded with stain. For example, here we have two samples that differ in the level of intact particles, as well as in the level of duplet and triplet formations. If one would keep track of these structures, the design space could be further detailed and the effect, if any, can be monitored. As mentioned in the previous slide, the level of internal stain can be measured. Here, the MiniTEM system has been used to image, detect, and differentiate between the two types of particle. This can be displayed as in the histogram to the right, where the internal stain measurement causes two distinct peaks, which almost gives a binary separation between the internal stain particles and the non-internal stain particles. As mentioned, as a comparative study, we analyzed two samples. One sample with only empty AAV particles, shown to the left, and another sample which, by our client, had been verified to contain genome-packed particles, shown to the right. The two different forms of internally non-stained and internally stained particles exist in both samples, and there is no significant difference between the samples. I will soon show this quantitatively. Analyzing the same two samples using cryo-electron microscopy, you can still see two different structures. One displaying a distinct outer shell and almost no internal density, characteristic of empty particles, while the two other structures displayed display an inner density with no distinct boundary between the shell and the core, characteristic of filled particles. Analyzing the two samples as well as a mixture of the two shows a clear and linear trend from 0 to 50% full particles using the cryoTEM. Analyzing the same sample using negative stain technique shows a stable level of internally non-stained particles. This means that the cryoTEM method is suitable for measuring the level of full and empty particles, while the negative stain method should be chosen when you want to assess particle integrity. Due to this, we always use cryoTEM when analyzing the level of full and empty AAV particles. We use our in-house developed software VAS to detect and distinguish between the full and empty particles. And for some of our clients' products, this method has been validated according to the principles of validation and analytical procedures ICH-Q2. So when working with electron microscopy, it is extremely important to choose the appropriate sample preparation and imaging method for your question of interest. If you are interested in the level of full and empty particles, you should go for cryo. If you are interested in the integrity of your particles, the level of purity and aggregation, you should go for negative stain. And from the experience we have with our clients, most of you would like to have information from both techniques. So to summarize, by having a MiniTEM system at your site, you would be able to do the detailed characterization of your gene therapy vectors and VLPs in early development, which can support both formulation and production. And by identifying critical and measurable parameters for your products in early development, and by using them throughout the whole process, you will be able to develop a robust production of high-quality products. And don't forget that we can be there to help you. Depending on your needs, you can use our services where we take care of the whole process. If you already have an electron microscope at your site, you can use our image analysis software VAS to generate data. And would you like to do all this in your own facility? MiniTEM is the way to go. Thank you very much for listening. And I think that we have some time now for questions, but you can of course also contact us directly. You have our contact information here. So thank you again, and over to you, Rita.
Well, thank you, Josefina and Martin, for your presentations. Before we get started on the Q&A, I'd just like to remind our audience that they can submit questions by typing them in the Q&A box, which can be found on the right-hand side of the presentation window. And we'll try to get to as many questions as we can. So here is our first question. So how many samples can the MiniTEM analyze per day? So what's the throughput?
Yeah, so as I mentioned earlier, it takes about, depending on the variability of the product being analyzed, it could take from maybe 20 minutes up to an hour of screening activity to get the ground data, image data, and then it could take either an additional hour for image analysis, or the image analysis is done directly. So there's also possibility to run the data crunching on a computer nearby. So about an hour per sample, so 8 samples a day would be an appropriate number to talk about. And from there, if you have easier samples, it could be more than that. Yeah.
Okay, thank you. So how many particle types are you able to detect and classify in a sample?
Well, this depends, of course, of the product itself. Usually, we can directly do distinction on different geometrical prototypes, such as spherical particles or elliptical particles, but usually biological particles are quite pleomorphic, and any such type of particle is detectable, of course. But then other geometrical classifications can be performed, but it's not just only the geometrical shape of the particles, it's absolutely the texture, the surface proteins, if they are interacting with each other, if they are placed in agglomerates or in aggregates, and these types of not primary particle features, but rather group particles, group dynamics can also be measured and detected.
Okay,
yeah.
Thank you. Here's our next question. So if you claim a correlation between particle physical characteristics, does that mean that bioassays could be reduced or eliminated?
I think that you can use it as a complement. We do this physically characteristic analysis, and our clients are then, with other techniques, looking into the biological features of what we see. And of course, if we can see a good correlation, then you can use electron microscopy instead of the biological assays. But I would not, totally take them away.
Okay, thank you. Here's the next question. So how many pictures do you need to characterize one sample?
Yeah, so again, it's up depending on the variability of the product. But usually when we screen AAV particles and we have a good on-grid homogeneity throughout the grid, maybe about 250 images per sample. And then it's always good to do a sample repeat, of course. So about 500 images, resulting in a couple of 100,000 particles being registered and analyzed. Yeah.
Okay, thank you. Here's the next question. Are these methods of measuring particle quality acceptable to monitor stability of the products and formally establish shelf life?
Let's see. I didn't hear the first.
Can you please repeat?
Sure. Are these methods of measuring particle quality acceptable to monitor stability of the products and formally establish shelf life?
Right. So we have done measurements tracking shelf life, and if we have correlated what we've seen in the images with other bioanalytics stating what if the product is stable or not. Usually what happens is the formation of larger aggregates which can be detected and measured.
Okay, thank you. Next question. Have you compared your results against analytical ultracentrifugation?
No, we have not. The assays that I know that our clients have compared our data with is ELISA and qPCR. And if they then after that compare those data with biological assays, I think that the qPCR and ELISA had an error of approximately 20%, while the electron microscopy analysis compared to the biological data had an error of 1%.
Okay, thank you. Next question. How accurate are the counts?
So the counts, just stating that we don't do transmission electron microscopy as a method itself is not so good for counting particle concentration in the sample, just to state that. But if we're performing an assay, and making an assay, then we would like the variability, or like the relative standard error, to be anyway. We're happy when we come down to 10% of relative standard error, and 10-20% is usually what we would expect on very detailed analytical questions. Comparing this with some bioassays, which are about in 20 to about 40%, it's quite stable results, I would say.
Okay, thank you. Does anyone ever validate the procedure?
Yes, we have validated some of our procedures for some of our clients and for their specific product then. Not all of them, and that is up to the client to decide if they want us to do that or not. Because they need to set the acceptance criteria. That is nothing that we can do.
Okay, thank you. Does the MiniTEM do cryo-EM?
No, the MiniTEM does not do cryo-electron microscopy. It is, well, first of all, it's not prepared for cryo-electron microscopy, and also going down to the MiniTEM is running on 25 kilovolt in acceleration voltage, and the interaction with ice and electrons with that speed is a little bit on the tricky side. So we haven't prepared the MiniTEM for cryo-electron microscopy yet.
Okay, thank you. Here's the next question. Full particles can be characterized into full heavy and full light. Does the cryo-EM or negative stain TEM detect different subtypes of full particles?
Yes, can you please repeat the question again, Rita? I'm sorry.
Sure, no problem. Full particles can be characterized into full heavy and full light. Does the cryo-EM or negative stain TEM detect different subtypes of full particles?
I guess that what you mean here is that you have particles packed with two different genomes, one which is lighter and one which is heavier. And we can sometimes differentiate between these, and it very much depends on the size of the genome. So it's not that I have a threshold that I can give you. This is something that needs to be tried out.
Okay, thank you. Here's the next question. So does sample formulation have an impact on imaging?
It can have. Of course, I mean, working with electron microscopy, we want to have as pure formulation as possible. But there is no problem working with, let's say, 10% of sucrose, even though we need to freeze it for cryo or use it for negative stain. But of course, if it's a really heavy salt concentration or sugar concentration up to, like, say, 20-30%, then we can have strange defects in the images. But that is something that you will see directly.
Okay, thank you. Can aggregate form during the TEM grids preparation?
Absolutely, it can. So again, I mean, using electron microscopy for measuring a concentration is not really optimal. It's a much better tool to, let's say, compare concentration of different particles from batch to batch to see if I have more or less aggregation in these samples.
Okay, thank you. So what causes the doublet or triplet AAV particles observed by TEM?
To be honest, we are not entirely sure. I've discussed this with many of our clients, and the hypothesis is that these are formed by particles that have been disassembled and then reassembled again.
Okay, thank you. Is TEM a method for characterization only or for release testing?
I would say that this is up to the client. I mean, we have performed validation of some of the methods where we know that the client need it because they use our data in clinical trials. And for batch release, I'm not sure if any of our clients are using our data.
Okay, next question. So what level of expertise is required for sample preparation, sample analysis, and data analysis using a MiniTEM?
Yeah, so when we set up a MiniTEM system, since we have all the expertise here at Vironova, we help out to transfer the sample preparation assay, and we train the personnel and staff at the site, and also usually we perform a initial test on the product before we have a system that comes over and being installed, so that we know that anyway we will have maybe an experimental version of an assay that we know that we can get good data out from, and then we help out creating these workflows, and then we train our clients to modify and to do QA on these workflows. Our experience is that our clients they like to have a starting point, and then usually they get their fingers quite dirty quite fast and want to experiment and try out things. And this is very simple. It's all workflow configuration is performed with graphical user interfaces and drag and dropping boxes for sequential work that we want the computer to perform and the microscope to perform. So, of course, we're biased here at Vironova saying that it is easy, but our clients say also that it's quite straightforward to use.
Okay, thank you. Here's the next question. Have you analyzed samples that were freeze-dried and reconstituted? So in your experience, what did you observe in regard to the enveloped virus?
To be honest, I can't recall that we have analyzed the viruses that have been freeze-dried. We have analyzed other type of particles that have been freeze-dried, but for viruses, no.
Okay, next question. So what about not completely full or not completely empty particles? Can your software distinguish them from fully full or fully empty particles?
It again depends on the size of the genome. I know that we have had one client which we were able to distinguish between two sizes of genome, and they were both characterized as filled. That was good. However, they also we have tested together with them some which were packed with a very short genome, and those particles they ended up in the empty peak, which the client was very happy because that was, let's say, not the right packaging. So it totally depends on the size of the genome.
Okay, next question. Can you use a particle standard with known concentration for viral particle quantification?
Yes, you can. Of course you can. It's a method that I would use very carefully, though. This because you can't be sure that the standard particle and your virus particle have the same affinity towards the grid. So it might be that they have, and then it will work perfectly well. If they do not have the same affinity towards the grid, you might end up comparing apple and pear and actually don't have a clue that you do so.
Okay. How was the quality of the negative stain preparation assessed?
Yeah, so going back to, let me show you the, where do I have that? I'm just trying to find this slide here, I think. Yes. So going back to this slide, a proper embedding of a virus particle. So the embedding, the embedding that is used with a heavy metal salt with negative stain is for two purposes. One of the purposes is to get contrast, because we have a carbon surface, and viral particles are made more or less of carbon. So it's carbon on carbon, and we wouldn't have so much of a contrast. So the salt is giving us a contrast shift on the particles, but also this salt is embedding the particles so that they are stabilized when we dry when the water is dried off. On the left images, on the left image, we see just a little bit of salt surrounding the particles, and the background is quite bright directly in the vicinity, or in the direct vicinity of the particles. Whilst in the right image, we have quite a more longer gradient of salt moving out from the particle, and we're measuring this type of curvature outside the particle, and with that we can assess then if the embedding is okay or not. So actually that is what we have on the x-axis here, x-axis here on the scatter plot to the right. So this is inbuilt into the MiniTEM software, and you just get this data out per particle, and usually we do this as an assessment per waypoint to see whether a location on the grid is okay for analysis or not. So this is something that should always be performed, and it's inbuilt into the analysis software. Yeah.
So, a related question, what's the best contrasting agent for negative staining for adenovirus?
For adenoviruses, we have tried out a couple, and I still think that the uranyl acetate is the best one.
Okay, thank you. Can you use immunogold to confirm and distinguish VLPs from debris?
Absolutely. And the gold particles, I mean, they are chosen very nicely, so you can very easily detect them as if you would like to.
Okay. So what's the typical turnaround time for sample testing?
I don't know if this is for the service or if this is for if you buy the MiniTEM. But I mean, for the service, normally you will have your results within 10 working days from the date we start working on them. And I mean, for the MiniTEM, you can run a couple of samples each day.
Okay. So how complicated is it to prepare the sample for MiniTEM measurements?
I mean, the preparation is rather simple. It takes like 5 minutes. You add 3 microliter normally of your sample to the grid. You blot it off, most of it with the filter paper. You wash it. Again, 3 microliter of water or buffer to the grid, and you blot it off with the filter paper and end up with 3 microliter of stain for a couple of seconds or a minute, and then blot off again with the filter paper and let it dry. That's the procedure.
All right. Well, thank you, Josefina and Martin, for your presentations and for fielding the audience questions. I'd like to thank the audience for their participation. And above all, I'd like to thank our sponsor for making today's webcast possible. Just a quick reminder, there are resources provided by our sponsor that are located in the green resource widget in the dock at the bottom of your screen, so you can go in and take a look at those. Today's program will be available for on-demand viewing through April of 2019. You'll receive an email from BioPharm International alerting you when this webcast will be available for replay. I invite you to forward that announcement to any colleagues who may have missed today's live event. Thank you for joining us, and have a great day.